Yeah, maybe that was too effective. But yeah, that's how your memory T and B cells will react. Once they encounter their antigen again, they go crazy. Anyway, so back to, so why do I cover memory T and B cells? Well, that's the whole thing vaccination tries to do. That's what we're trying to do is build up your reserves of memory T and B cells so that when you encounter the actual pathogen, that your body is able to mount a faster response and more efficient response to combat that infection before it takes a hold in your body. Instead of your whole body getting infected, then you have to wait for all your dendritic cells to process the well, one, your neutrophils have to try to help neutralize the pathogen as much as they can. Dendritic cells have to uptake all the antigens, present it to naive T and B cells, and then they have to screen all these naive T and B cells, and then they have to do a further selection to make sure that they are recognizing the same antigen without attacking your own body, and then they finally can fight off your infection. That takes time. So save yourself time by getting a vaccination. So I know this is very complicated. Am I expecting you to know all of this? But not really. So what's happening here is like, okay, what you're doing is introducing the antigens from a pathogen into your bloodstream. And again, here we have our dendritic cells, and they're hopefully going to present the antigens from the vaccine to the rest of your immune cells. So here we have a live virus vaccine or a DNA vaccine. So what are you going to, or inactivated, or sometimes we call it attenuated, where it is the live virus, but you process it in a certain way where it can't replicate as much, or sometimes it can't replicate at all. So again, what's happening here, hopefully those dendritic cells upstream here start to activate the naive T cells. Again, most of them become effector T cells, but small subpopulations. So again, become naive T or become memory T cells. So rather than the effector T cells. So again, the effector T cells, they go off to battle and die, whereas the memory T cells, they hang around. So when you get the same infection again and the actual pathogen, then they can be effector T cells, or they can continue to make more memory T cells. All right, so let's talk about polio or poliomyelitis. So again, myelin, what is that, or what does that cover? So remember, if you were here last semester, myelin is what covers those myelinated axons on neurons, right? So poliomyelitis, the thing about polio, it attacks the cells that make myelin. So what often happens is, and actually attacks nerves. So look at these poor children. What's happening here? Well, they have infection with polio and poliovirus. So they have poliomyelitis. So you notice that, look at the muscles right here. These poor children, they actually have atrophied muscles. And why? If you have severe poliomyelitis, that can actually cause degradation, death of motor neurons. Remember, motor neurons are the ones that cause skeletal muscles to contract and move. So if you have a total death, or if those motor neurons die, can you really have, so what happens when you have neuron damage? Typically when you have neuron damage, that results in atrophy of the affected muscles and also affects the contraction of the muscles as well. But if that nerve is completely denervated from that skeletal muscle, you don't have any nervous signals, you can't control that muscle, and the muscle atrophies like you see here, these poor children, right? So yeah, oh my god, yeah, so look at this. Does she have any calf muscles? And look at her thighs. Yeah, so her thighs and all the calf muscles here and all the quads and hamstrings here, probably this poor girl, her motor neurons have been attacked by the polio virus. So this is why, so what happens if that nerve that innervates your diaphragm dies? Well, this is why back in the day, yeah, so I should check if he's still alive, but this is the last man in America who needed something called an iron lung. And yeah, polio used to be such an epidemic that they needed all these iron lungs. You won't find these in modern hospitals these days. But what did the iron lung do? Well, it used, remember, Boyle's law? As you increase volume, as you affect pressure, volume pressure are inversely proportional. So if you're able to, so what happens here, they actually used to regulate the pressure inside this chamber. So by lowering the pressure in the chamber, this allows the lungs to expand. And if you increase the pressure, that causes the lungs to collapse. So this was actually doing the breathing for these people. Why? Because if the diaphragm is denervated due to poliovirus, these people can't breathe on their own. So this is why we need all these iron lungs back then. So that's why, so who is this person? This was Jonas Salk. And what amazing thing did he do? He invented one of the first polio vaccines. And what he did is like back in 1954, he wanted to prevent this. So again, what happens? So he invents the vaccine, and by 1962, from 45,000 people, he dropped to 910. So this is an amazing thing. He never patented the vaccine or earned any money from his discovery. Yeah, he really believed against preventing this horrible disease from happening to people. He just wanted to invent it and distribute it as widely as possible. And I know, like, a lot, that's funny because, like, I think 1 or 2 years ago, I had someone who was an anti-vaxxer in my class, and he's like, oh, the pharmacological industry, and they're making money off of it. Well, one, if you look at yes, the pharma industry needs to be controlled. They do charge too much for drugs. I mean, it's ridiculous when you see what we pay compared to other countries. So I wish we had more funds so that we could have more Jonas Salks. But this is amazing what he did, and I wish we had a society that was supporting the development of free vaccines like this. But again, that's a big problem of his, yeah, the pharmacological industry. It's all about stockholders. So how did he invent the polio vaccine? So what he did is take the polio virus, but he didn't just inject it into people. What he did is actually pre-treat it with formaldehyde. So formalin, also known as formalin. So the thing about, or solution is called formalin. So the thing about formaldehyde, what does it do to proteins and DNA? So it does something, again, it's kind of chemistry, organic chemistry, biochemistry. It does a process called cross-linking. So what it does, is actually causes proteins and DNA to kind of shuffle around its chemical bonds. So what else do we use formaldehyde for in science? Well, preserving. Remember those fetal pigs? So notice that, again, are those fetal pigs they still have the shape of a pig, but are they going to grow up into adult pigs and have little fetal pigs of their own? No, they have the shape, but they're inactive. They're dead, right? So this is what he used to quote unquote kill these polio viruses. Again, viruses, they're kind of like, are they alive or not? They're technically not alive because they can't replicate on their own, but by treating them with formaldehyde, they still have the shape of poliovirus, but since all their proteins and DNA have been chemically bonded to each other, it's not really able to replicate and be active like a normal live poliovirus. So now we have the overall shape of this poliovirus. What can happen? Remember, your dendritic cells can take this poliovirus, take the antigens, and the thing is that because it's inactivated with formaldehyde, it shouldn't replicate and cause all those awful symptoms with poliomyelitis. So your immune system is going to train itself to recognize all these surface proteins on polio or some of the internal proteins. And it's going to train your immune system to recognize polio, all the protein and DNA components of poliovirus. Now this is related to another vaccine. So